Hello, my name is Julianne and I'm one of the authors of the paper Neurovascular Phase Coherence is Altered in Alzheimer's Disease, which is published in Brain Communications. Alzheimer's disease is the commonest form of dementia and it's characterized by neurodegeneration, amyloid-beta plaques, and tau tangles. Recently, however, the role of the vasculature and its connection to the rest of the brain via the neurovascular unit are being highlighted as important for disease progression. The goal of our study was to quantify the changes in the neurovascular unit in vivo non-invasively. For a more comprehensive understanding of the regulation of blood flow in the brain, we first start by considering the cardiovascular system. It consists of the heart, the lungs, the blood vessels, and the blood. Blood flows continuously around the closed system of vessels and delivers oxygen and nutrients to cells. This flow is finely regulated, involving different oscillatory processes. Let us consider the journey of red blood cells to the capillary bed. The main function of a red blood cell is to transport oxygen to be used in the production of useful energy in the cells. First, oxygen is collected from the lungs by deoxygenated red blood cells. The average breathing rate is around 15 times per minute. Then the heart pumps the cell around the rest of the body, and the heart beats around 60 times per minute. The heart and lungs are examples of biological oscillators that give rise to rhythms. The velocity of flow is regulated by many local oscillatory processes, in addition to the heart and respiratory rates. In the arteries and arterioles, rings of smooth muscle cells adjust the flow by contraction in response to pressure, called myogenic activity. This vasomotion involves oscillations in the diameter of the vessel wall, which usually occur around 6 times per minute and can be observed in blood flow signals. The nervous system can also influence blood flow. Nerves surrounding the vessels can signal to the muscle cells, causing even more vasomotion. Further, vessel walls are aligned with a thin sheet of endothelial cells. One of their functions is the regulation of blood flow by rhythmic release of substances that cause vasomotions. This is one of the slowest rhythms in the cardiovascular system. Nutrients and oxygens are rhythmically delivered to the cells from the smallest blood vessels. The capillaries are very narrow, deforming the red blood cells and thus helping to release the oxygen. Biological oscillations do not stop once the oxygen is delivered. They can also be found on even smaller scales. Within individual cells, oxygen is used in energy metabolism to rhythmically produce the energy-carrying molecules known as ATP. This cycle happens around every 2 minutes. The further the blood flows from the lungs and heart, the more oscillations become involved in the regulation of blood flow. In addition to these processes, in the brain, the neurovascular unit is involved in the regulation of blood flow. Here we show an illustration of the neurovascular unit. Notice how astrocytes link the blood vessels and neurons. Alzheimer's disease is associated with a leaky blood-brain barrier, inflammation, neurodegeneration, amyloid-beta plaques, and tau tangles. We work towards a non-invasive approach to evaluate the health of the neurovascular unit non-invasively in humans. Our assessment is based on recordings of brain oxygenation, neuronal and cardiorespiratory activities captured by functional near-infrared spectroscopy, EEG, ECG, and respiration effort, respectively. The time series were analysed using methods that can capture multiscale and time-varying oscillations. These techniques aim to characterize the time-dependent, finite time dynamics of biological systems and include the wavelet phase coherence and methods available in the toolbox MODA. Our results are based on 25-minute continuous recordings from 39 participants, 19 of which had Alzheimer's disease and 20 that were control participants. Our results show decreased oxygenation power in the neurogenic and myogenic frequency bands in Alzheimer's disease participants compared to controls. However, a lot of our interesting results are not in the power, but in the interactions, and we show decreased neurovascular phase coherence in the neurogenic and myogenic frequency bands in Alzheimer's disease compared to control participants. Quite unexpectedly, we also detected an increased respiration rate in participants with Alzheimer's disease. This discovery promises to open up a whole new world in the study of Alzheimer's disease, and it most likely reflects an inflammation, probably in the brain, that once detected may be possible to treat, so that severe states of Alzheimer's might be prevented. In conclusion, Our approach enables Alzheimer's disease to be detected simply, non-invasively, and inexpensively. For further details, please see the full paper in Brain Communications.